Hello, everyone. My name is Megan Kirk, and I'm an East Tennessee State University alumni. I wrote my honors thesis on neonatal life-saving technologies. I would like to take a moment and thank my co-author, Dr. Marco Kostick, my mentor, Dr. William Blanton, and my proofreader, Dr. Joseph Schrift. They have each made a huge impact on my life and in furthering my career. Babies are considered full-term at 39 weeks. Anything earlier than this is considered preterm. It is common for most preterm babies to have issues, but their biggest struggle is with their heart and lungs. Babies born before 24 weeks usually have less than a 50% chance of survival, but as weeks progress, fetal development enhances as well as the chance of survival. The simplest of tasks for full-term babies and even adults is breathing. Breathing is a thoughtless act. We don't have to tell ourselves to breathe or to expand and compress our lungs, but for a premature baby, what may seem so thoughtless to us can be impossible. Preterm babies struggle to breathe so much due to their underdeveloped lungs. Here we can look at biweekly fetal development, specifically of the lungs. In the organogenesis phase of fetal lung development, we have the embryonic and pseudoglandular stages, where major airways begin to form. Then we progress into the differentiation phase, where we have the canicular stage. This is where cell specialization occurs and the air-blood barrier begins forming. Then we get into the saccular stage, where the air sacs of the lungs begin to form. During this stage, we can detect the first traces of natural surfactant in the amniotic fluid. Lastly, we transition into the alveolar stage, where air sacs begin separating into smaller air sacs to gain more surface area until birth. So around 25 weeks is when surfactant is first detected in amniotic fluid. Surfactant is an surface active substance that works in the lungs to provide surface tension to avoid lung collapsing at the end of exhalation. Surfactant is a major benefactor in assessing the chances of survival of a preterm infant. If the preterm baby is far enough along in development that they can produce their own natural surfactant, they can be treated with additional artificial surfactant that will increase their chances of survival without having any complications later on in life. In this table, we can see the correlation between gestational age and the likelihood of survival. As we can see, as the weeks progress, so does the likelihood of survival. The overall objective of this study was to engineer new technologies that would ultimately decrease infant mortality, specifically after preterm birth, by looking into what could be done with the fetus while still in utero, as well as after preterm birth. Preterm babies receive care in a NICU, which stands for a Neonatal Intensive Care Unit. In the early 1900s, hospitals were very wary to adopt the first incubators during this time. They used wooden boxes with warm water bottles because that was all they had. And hospitals deemed this as too barbaric during that time. However, a French obstetrician, Dr. Coney, saw the need for this innovation to be brought to light. To increase its popularity, Dr. Coney would offer his incubator services to premature infants free of charge. He would show and tell these infants in their incubators at local fairs and shows, charging onlookers 25 cents a piece to see the technology keeping these babies alive. Ultimately, Dr. Coney's plan was successful, as shortly after, hospitals began adopting incubators into their NICUs in the 1950s. A few years later, the appearance of incubators evolved immensely into an all-glass casing with side doors, allowing easier access. This was leaps and bounds from the original incubator. Presently, not only do we have a highly developed, fully functioning monitorial incubator, but a number of other devices and machines are used on a daily basis in the current NICU. So why does this research need to be done if the current protocol isn't broken? There are still so many questions that have not been answered regarding fetal lung development, and learning the whole lung developmental process could possibly answer some of these questions. Is there a way to stimulate the secretion of natural surfactant prior to 25 weeks? Would the fetus even be able to utilize surfactant prior to 25 weeks, or would their lungs still be too underdeveloped? Can we jumpstart or manipulate the fetal lung developmental process in a manner to allow the fetus's lungs to mature quicker? Can researchers cultivate a precautionary drug that high-risk mothers can take that would rapidly accelerate fetal lung development in case she would happen to go into preterm labor? There is always room to improve, especially in the medical field where new questions are constantly being presented. This type of study is needed so we can save more lives. In conducting this study, I've researched past as well as current studies as well as their results. I've also asked myself what methods haven't been tested and what questions still haven't been answered. Here's what I found. The first thing I looked into was artificial surfactant supplements for the mother to take while the fetus is still in utero. Similar to prenatal vitamins, this supplement would target and jumpstart the production of surfactant before the fetus produces its own natural surfactant at 25 weeks. So, what exactly is surfactant? Lung surfactant is a complex with a unique phospholipid and protein composition and is secreted by type 2 alveolar cells into the alveolar epithelial lining. A similar study has been previously conducted where mothers went through several rounds of corticosteroid injections between 22 and 26 weeks to hasten fetal lung development between weeks 28 and 34. However, there is no evidence to suggest that the corticosteroid injections may or may not enhance fetal lung development if administered prior to 23 weeks. The steroids are used to speed up the secretion of natural surfactant, but what would happen if artificial surfactant was also administered to the mother prior to 23 weeks? Independently, they yield good results, but could coupling these together better prepare the fetus for premature birth? Again, would the fetus even be far enough along in its lung developmental process for this to be beneficial? Could lung development possibly be stimulated week by week or even stage by stage? Could artificial surfactant be injected directly into the amniotic fluid near the fetus's nose and mouth to boost lung production, or would this be too risky? If it would be beneficial, what type of artificial surfactant would be used? Would we use porcine, bovine, or synthetic artificial surfactant? All of these relevant questions that are still being studied, among more. The last technological innovation that I looked into and that is still being studied is an artificial womb or a biobag. It is a large clear bag filled with artificial amniotic fluid that is continuously being exchanged to maintain and preserve the baby's fluid-filled lungs. After a premature birth, the baby would be placed in an artificial womb, being constantly monitored but not exposed to the outside environment, where they would continue on with their development just as they would in the mother's uterus. In 2017, this study was conducted on lambs. 8 out of 10 lambs survived and grew inside an artificial womb for 4 weeks, which was the longest any animal had done so at this time. The lambs developed just as they would have in the mother's uterus. All 8 lambs grew into adulthood, seemingly having no problems. However, it is difficult to attain the grasp on where the lamb stood mentally, as there's not an intelligence test for lambs. This little bit of information still makes researchers question the biobag's effectiveness, as they cannot understand how this would affect a human fetus mentally. The idea is wonderful, but further research is still needed in order to call the biobag a success. With every new idea, however, also comes coupled with drawbacks. Will new drugs have any long or short-term side effects for the mother or child? Are the healthcare system and researchers willing to perform experimental trials on pregnant women and their unborn children? Is any of this even ethical? There will always be drawbacks with every idea, but it is all on a weighted system where we hope the advantages outweigh the disadvantages. All in all, I'll leave you with these still unanswered questions. If artificial surfactant is injected into the amniotic fluid before the fetus is 25 weeks, will the fetal lungs develop faster? If the mother could take artificial surfactant supplements, would this yield the same effect? Can corticosteroid injections be beneficial if administered prior to 23 weeks? Could artificial surfactants and corticosteroid injections be coupled together to yield better, more efficient results? And lastly, can the biobag successfully serve as an artificial womb for the human fetus, leaving no mental drawbacks? All of these ideas warrant further research that may eventually surpass the current practices in neonatology and save lives worldwide. Again, I am Megan Kirk, and I'm an East Tennessee State University alumni, and I would just like to thank you for your time.